Welcome everyone to Ecology Live. I'm Mark Cadotte. I'm editor of the BES journal Ecological Solutions and Evidence. If this is your first time joining us, welcome. And welcome back to all of our regulars for this series, which is now in our 7th talk. You can check out who is still yet to speak on our website. And while you're there, why not sign up to receive our newsletter and find out more about everything else the British Ecological Society does. It is my great pleasure to introduce Tadashi Fukami for today's talk. But first, we would like to thank Wildlife Acoustics for the wonderful soundscape that started this session. We'd also like to thank Oxford University Press, who are sponsoring this whole season of 12 talks. You can find details of the Ecology Live reading list, which includes discounts and some free content, on the slide at the end of today's talk. Just a note, this talk is being recorded and will be added to our YouTube page afterwards. Also, people can submit their questions via the Q&A box throughout the talk. There's no need to wait until the end. There will be a short Q&A session at the end of the talk. Your questions can be named or anonymous, you decide. You can also upvote any questions you find particularly interesting by clicking the thumbs up icon to bring it to our attention. Now, let me introduce my good friend, Tadashi Fukami. Tad Fukami is a professor in the Department of Biology at Stanford University. He has greatly advanced our understanding of ecological contingency and the role of priority effects in shaping community composition and diversity. Because of his profound contributions to community ecology, Tad was named as a Fellow of the Ecological Society of America and was given the Presidential Award from the American Society of Naturalists. Today, we're very fortunate to have Tad speaking to us, and he'll be taking us to a microscopic world where flowers are islands. Over to you, Tad.
Thank you very much, Mark. Let me share my slide now. Is it working now?
Yes, looks great.
Okay. Well, thank you very much, everyone, for coming to this talk. I'm very honored and excited to share some of the findings that we have on flowers as microbiome. And so islands have played an important role in ecology to think about evolution of species and structuring of ecological communities, not just to understand islands, but generally to have general principles about ecological dynamics. And I think one of the most famous one, as most of you know, is the MacArthur Wilson's theory of island biogeography, where they say that to understand species diversity on an island, you look at immigration rate and extinction rate, and then you can have a good prediction about how communities are being assembled on islands or any kind of local habitat. But a small but increasing number of studies have also shown that it's not enough to think just about immigration rate, because immigration history can be important. For example, if you have different islands that are identical in chemical and physical conditions, if you have this, even if you have the same species coming to these islands after some major disturbance, for example, by having some variation among local habitats in terms of the history of immigration, who gets to the island first and who gets there later, that differences in immigration history can end up to create different ecological communities with different species. And this is what I'm going to refer to as historically contingent community assembly. And this happens through what we often call priority effects, where early arriving species, often these are the early arriving ones just by chance, they change the local conditions in a subtle way, often that we don't realize, but because they make some changes, how species are going to interact through competition, predation, mutualism, different local species interaction are going to depend on who were the early arriving species. And because of this increasing evidence from theory and empirical studies showing that the contingency can be important. That led to this famous quote from John Lawton in Oikos 1999, saying that the community ecology is a mess because of this contingency, including historical contingency, we can't really talk about general ideas or general theories in community ecology. And that bothered many community ecologists. But one way to move forward, make progress, is to ask this question: when should history matter? We might not be able to understand exactly how history is going to affect any specific community, but if you have a good understanding of what ecological conditions make history community assembly more sensitive to history, that could give us some good understanding of community assembly. And to address this question, I wanted to introduce flowers as islands, habitat islands, where microbes like yeast and bacteria grow in nectar. And shortly after arriving at Stanford, over the past 10 years or so, we have been going to this field local site called Jasper Ridge Biological Preserve. And I was working with many good people working with me. Those are some of the people, PhD students, postdoc, and some of them are visited from other places like Spain, Canada, and Japan. And what I wanted to do today is to give you some examples of what we have found in relation to historical contingency. But before that, I wanted to give a brief introduction to this island system and why this is a convenient system for thinking about priority effects and community assembly. I have 5 reasons here. Just like any microbial communities, if you have bacteria and fungi like single cell fungi like yeast, that's a convenient system because they have short generation time. And so you can look at multi-generational community dynamics over to humans, to us, in a very short time. So that's true with any other microbial system, but what's unique about this system is that we only have a handful, small number of species living in nectar. So it's a simple system, presumably because there's so much sugar that the osmotic pressure gets really high. So only a handful of species of bacteria and fungi have evolved to withstand that harsh condition. Another thing is that the nitrogen is very limited. There are a bit of amino acids in nectar, but because it's a nitrogen limiting environment, that seems to be another reason why there are only a few species that can live. So that means that unlike many other microbial communities, which are very diverse, we have a simple system. So we can begin to tease apart who is playing a role, who is playing what role, and how they're interacting with one another. So it's a good experimental system. Another thing is that to do experiment, we have to be able to culture them. And not many microbes, of course, can be cultured at the moment. But in most of the common species that we find in nectar, they are culturable. So we can isolate the different strains of microbes and do experiments both in the lab and also in the field. Another thing is that, so this is related to the title of my talk, it's a well-defined habitat, flowers, which is not always the case with any communities. If you go to forest and want to study tree community, how to define your community, that gets difficult and not trivial. But in this case, because those microbes that we see in nectar are found only in nectar, mostly, except on bodies of pollinators who visit those flowers, we don't see them any elsewhere. So these flowers are truly conceptually a good island system so that we can focus on what happens at the different local communities. And then if you deal with microbes, the challenge is that we can't really see them. So where they're going, we want to understand immigration and disposal. But in this case, what's convenient is that the way that these microbes get around from one local community to another, which is flower to another, is via hitchhiking on pollinators. In our case, with our native plant in California that we study, the hummingbirds are the main one, but we also have bees. And so if you look at flowers, and if the hummingbirds go to one flower and that happens to be an old flower full of a lot of microbes in nectar, and if that hummingbird then go to a new flower, which is fresh nectar, free of microbes, then that's how the disposal happens in this system. And so by keeping track of where hummingbirds go, and sometimes bees, you can have some idea of how how immigration happens in this system. And so now that I introduced this system, I wanted to show you mainly just two experiments because we don't have too much time to tell you what kind of experiments you can do to ask this question that I posed: when should history matter with this experimental system? So one experiment that I wanted to briefly talk about was done some time ago, and it involved 4 species: 2 species of yeast, Mechanocobia, Stamarella, and 2 species of bacteria, Gluconobacter, Acaya. Those are species that were all isolated, that we isolated from our monkey flower nectar. And so in this experiment, we wanted to just do a lab experiment. And so instead of using hummingbirds, we used pipette and PCR tubes as our artificial flower, and then just playing with the order of introduction here. And so, for example, you can introduce 2 species of yeast on day 0 and wait it for 2 days, and then introduce 2 species of bacteria and see what happens after a while, after 4 days, which is about the same as the longevity of the flowers that we see in the field in this species. You can flip the order to look at the effect of arrival order and see what happens. You can also put all of the species together and see what happens if you have a simultaneous introduction of species. And so I'm going to quickly show you what we found in, this is a paper in 2014. And so it was a 4-day experiment, but you can also transfer a little bit of nectar after 4 days and keep transferring nectar to a fresh nectar, kind of simulating what happens in the field where you have a flower and microbes coming from old flower to new flower, and if that process continues. If we introduce all of the 4 species together, 2 species, 1 yeast and 1 bacteria, were the ones that persisted. And that made sense to us because those are the most common yeast and bacteria respectively in the field in real flowers. But if you use the sequential introduction, what happens is that if you have yeast first, then only one of the yeast species, the dominant one, Mechanocobia ricoiferi, was persisting. All other species went extinct after a while. And then if you use the bacteria first treatment, then it's only the Gluconobacter, also called Neocomagatia. That's the only one that persisted. So already we see a very strong effect of introduction order or priority effect. And so we have some additional data that gives us some idea of what happened. And it turned out that the pH and amino acids seems to be the key in understanding how this very strong priority effect happens. It turns out that the yeast is very good at growing under very limited amino acid concentration. We have a genetic, and now we did the genome sequencing of this species and see that there's a lot of genes that are responsible for taking up a lot of amino acids very quickly, but they are not very good at tolerating low pH. On the other hand, if you look at bacteria, they're not good at surviving under low amino acid concentration, but they are very good at surviving even if the pH is really low, like almost 2. And it also happens that this is a change that the yeast would do. So they take up a lot of amino acids, but they don't change the pH as they grow in nectar. And it's the opposite is true. And this is an acetic acid bacteria. So they acidify nectar a lot and change the nectar pH and lower pH dramatically, so dramatically that the yeast cannot survive, but the bacteria themselves can be there, even though they don't change amino acid concentration as much as yeast does. And so you can see it's a simple scenario here. Basically, each of them is making nectar conditions more favorable to themselves than to the other. And if you put these 2 species together, this is the change that we see, meaning that the coexistence can happen. And by the time this change happens, it's already the flower is getting old anyway, so they have to move to another flower. And by keep going to the new flowers, they can probably persist at the meta community scale. So this was all done in the lab, but we all the ultimate goal is to understand what happens in nature. And so encouraged by this interesting results, we went out to different places around San Francisco where our university is located. We are near San Francisco, and went to different places to get lots of as many nectar samples as possible. And what we found is that this pattern, so each dot represents an individual flower here. You can see that if you have a flower with lots of yeast growing, then we don't see bacteria. If you have a flower with lots of bacterial growth, then we don't see yeast. So this L-shaped pattern is consistent. It's not a direct evidence, but consistent with this idea that there's a strong priority effects happening, so that it's a mutually exclusive possibilities, either yeast or bacteria in most cases. And there's some evidence also showing that in a completely different place. So there's a study done in Japan in a different plant species, but we also see the same kind of L-shaped pattern in that system too. So it's not just unique to our monkey flower plant in California. And so this is just a summary that I have here. The yeast and bacteria seems to have a inhibitory priority effects happening. And we have Acinetobacter also as another common species that we see in our plant as another bacterial species. But we are not really, we are microbial ecologists, but we also wanted to see whether this priority effects has any functional consequences. And so to look at that, we also looked at what they might do to the plant pollinator mutualistic relationship. So we did a series of, we've been doing a series of experiments. For example, we can use artificial flowers and with different kind of nectar chemistry put into these different flowers and artificial flowers and see whether hummingbirds have preference. And we also did some experiments, the similar kind of experiments with honeybees, because we didn't want to just focus on hummingbirds, but we also wanted to see the results that we get also applies to bees. So here, again, we have a nectar, artificial nectar with, for example, you can manipulate pH, or you can manipulate sugar composition or amino acid concentration, different things that we might think that they might affect the pollinator preference. And what we found is that through these experiments, what we found is that the yeast doesn't really have an effect on hummingbirds, and sometimes a slightly positive effect on honeybees and bumblebees. Other people looked at bumblebees too. But if you have bacteria, you can experimentally introduce bacteria or change pH to simulate what happens to when you have bacteria growing in nectar, then they have a negative effect in the sense that the hummingbirds didn't have a higher preference for yeast-dominated or clean nectar compared to bacteria-dominated nectar. And we also looked at the flowers themselves, and we found that the pollination, the probability of pollination, and also the number of seeds produced by flowers are also negatively affected by bacteria. And so when we look at the priority effects, what happens is that the priority effects can have maybe an indirect impact on plant pollinator mutualism, because, for example, if the flowers get dominated by yeast, then through effect on bacteria, they might have a positive influence on plant pollinator mutualism because they keep the bacteria in check. So they suppress bacteria that have a negative effect on plant pollinator mutualism. So there are a lot of functional significance of this priority effects happening in microbial communities. Getting back to this result, I wanted to also show you what happens when we have a fluctuating temperature conditions. In California, we have a wide daily fluctuation in temperature because we have a Mediterranean climate. And so we wanted to compare what happens under constant versus fluctuating temperature. And you can see that in both places, cases, there are strong priority effects, but relatively speaking, because under fluctuating condition, there's some species coexistence allowed, we can say that under constant temperature, they have a very strong priority effects, and that changes if you have a fluctuating temperature. It's a relatively weak priority effects in the sense that sometimes multiple species can coexist. And the reason seems to be that, so this is what I showed you under constant temperature, but the reason why we have the difference between constant and fluctuating temperature seems to be simple, that under fluctuating temperature, in this case, everyone is sort of struggling to grow. And so the growth is slower, which leads to the higher probability of species coexisting. And so getting back to this question, when should history matter? It was just a small experiment that we did, but I think potentially you can say that from the temperature experiment results, you can say that maybe one potential answer could be that when the environment is less variable, that may be when we should think a lot about what history does to ecological communities. I have one more experiment I wanted to talk about, and this is unpublished data, but I think it's an exciting one, so I wanted to share, even though it's not published yet. And so this experiment led by one of my current PhD students, Kelly Chappell, who was interested in thinking about rapid evolution and the effect of evolution over a short time on priority effects. And so here we have under the lab conditions again, we have this one strain of Mechanicoria ricofia yeast isolated from our monkey flower nectar. And in artificial nectar, which we just did a simple disposal by pipette in the lab every other day for 2 months. And it's a simple transfer of the population. And at the end of this 2 months growth, we have what we call evolved strains. And since then, after this experiment, Kelly did genome resequencing of these strains. So we have evidence that there are actually some genetic changes that happened over 2 months. And by the way, this 2 months roughly corresponds to the flowering season of our monkey flower plants in the field. And this experiment, this transfer for 2 months transfer was done in 3 different conditions. One is what we call normal nectar, which is the nectar with lots of sugar and amino acids that are similar to the actual nectar that we see in the field. But we also had another treatment where we had a bacteria grown here in this nectar, but filtered out so that we can have yeast growing under a bacteria conditioned nectar. Everything else the same with the normal nectar treatment. And then we also had a low pH nectar. So this is the same nectar as the normal nectar, except that we changed pH so that we thought that that's the key factor that bacteria changes that might have influence on how good yeast, how well yeast grow in nectar. And so after with this 2 months transfer, now that we have these strains, experimentally evolved strains in our freezer, we can use those strains to do classic priority effects experiments. And so, for example, by using Acinetobacter as the potential competitor against yeast, we can do experiments that's a simple one with flipping arrival order and see what happens to quantify priority effects. But because we have other strains that we prepared, then you can do the same experiment, but using evolved strains. And we can also do the same thing with those strains that are evolved in bacteria-conditioned nectar or low pH nectar. And I wanted to share some results here. And so we want to compare these different strains, all the same species, but have gone through some short period of evolution for 2 months. And if we grow them just by themselves, in normal nectar, this is what we see, that the density is not very different. And if anything, for some reason, the strains evolved in low pH nectar didn't grow as well as the other strains. But then we can also look at what happens if we make them grow with bacteria. So this is what happens if you have bacteria as the initially dominant species, and so they're probably going to be this antagonistic interactions. But what was interesting here is that compared to what happens with ancestor, the difference between these two, the monoculture growth and the multi-species growth, that difference was different among these strains. So if you look at ancestor, there's a big change, and that's we expected that because we've known that there's a negative priority effects between yeast and bacteria. And it's the same here. But if you look at low pH nectar and bacteria conditioned nectar, that difference that quantifies the strength of priority effect, the negative priority effects is smaller. And we can statistically confirm that. And so basically what we find is that it was just 2 months, but the 2 months evolution over 2 months seems long enough to see some genetic changes that resulted in differences in strengths of priority effects. And so getting back to this question again, when should history matter? Based on this experiment, I think the potential answer, which can actually potentially general in this case too, even though it was just one experiment with our nectar yeast and bacteria, the answer is that when species are evolutionarily naive, when they don't have a recent experience with having to deal with the potential competitor, that might be when priority effects can be very strong. And those are maybe the cases where we have to think about historical contingency. So it was 25-minute talk. So I have many more experiments I wanted to talk about, but I don't have time. So what I wanted to do is to briefly go through some other answers that we came up with. So we did another experiment with the nectar yeast, 6 species of yeast. There are 6 other species that we see in monkey flower nectar. And we found that if you look at the phylogenetic relationship between the species, then when we have a closely related species in our species pool, that was when, based on this experiment that we did about a decade ago, is when we found a lot of strong priority effects. Another experiment we did, we measured a lot of ecological traits of these bacterial and yeast species, and what we found is that you can actually use niche overlap and niche impact and requirement, those things about ecological characteristics, to have some predictions about historical contingency. We also did a field experiment at Jasper Ridge, and what we found through exclusion of pollinators and bees is that if you have immigration rates that's frequent, that seems to be the case where history is important. And as a final experiment, I wanted to briefly mention, here we did the idea called modern coexistence theory to come up with some predictions based on equalizing destabilizing forces to think about when history matters. And so this is a very busy slide here, but I'm just putting all of the things that I have here. So when you look at this slide only, there's nothing about nectar microbes. It's potentially, our hope was that by doing experiments with the nectar microbes and flowers as islands, we can come up with potentially generally applicable answers to this overarching question, when should history matter? And so this quote that I talked about near the beginning of my talk, I think this was a good statement that stimulated community ecology. But actually, the quote that I like better is this one, more recent. Diane Srivastava said that ironically, it's perhaps in chasing contingency that we may discover general truths about mechanisms of community ecology. So contingency seems like a challenging thing to understand communities, and it has obvious direct implications for applied issues like ecological restoration, and also how to manage gut microbiome for health and so on. So there are a lot of applied things that we as community ecologists can contribute to, but I think instead of moving away, shying away from thinking about contingency, we can directly study them, particularly if we have island system like flowers for experimental system, so that we can have some general principle that might be also applied to other communities like plants and animals. And so if we want to know more about historical contingency, I have this review article that I hope is useful for you to learn more about historical contingency, but that's now 6 years old. And so the field is moving forward, and there are more and more studies coming out on priority effects, which to me is very exciting. So to give an update on the current status of knowledge on priority effects, I'm working with Ben Delrief in Germany and many others around the world to organize a working group to look at the what's available now, what new knowledge is available now in relation to historical contingencies, not just the microbial communities, but all, just generally in any community, any type of communities. And if you want to know about flowers as islands, I would suggest that this concise review article that we wrote a few years ago, Kelly and I, there is a review article that can be hopefully useful. And then there's also a concise article that we read, we wrote, led by Sergio Alvarez Perez. That could be interesting. And I'm happy to communicate with you also through Twitter. So that's all I wanted to talk about. Thank you for listening. And if we have some more time, I'm looking forward to trying to engage with you and answer questions that you might have. Thank you very much.
Thanks a lot, Tad. That was a fantastic talk and a really great overview of what is a very powerful experimental and observational system for answering some very fundamental questions in ecology. We do have a bunch of questions coming in. I've been looking through them. There's a lot of really good ones, but maybe I'll try to get through 5 or so just in the time that we have. The first question comes from someone at ETH Zurich. And you sort of touched on this when you're going through your other questions, when you talked about stabilizing, equalizing mechanisms. So this person is asking about priority effects and the relationship with coexistence theory. And so I was thinking, as you were talking, is there feedback for priority effects to better inform coexistence theory? How can we bring this into coexistence theory more thoroughly?
Yes, yeah, good question. I think that historically the focus, main interest has been to think about this question of whether do species coexist or not. And in the case of non-coexistence, exclusion, there has been a focus on deterministic competitive exclusion, where regardless of the history of community assembly, it's always the same species that's going to win. But it turns out that the theory is already relevant to priority effects, because there's when you look at the region of possibilities that are predicted by this modern coexistence theory, there's this other side where you can also, theory seems useful in thinking about historically contingent competitive exclusion, where, so that's the case where priority effect happens, that it depends, there is a exclusion predicted, but which species are going to win depends on which came first. And so the other side of the modern coexistence theory that already was there, but not looked at so much, because everyone was looking at deterministic exclusion. The theory is actually available for us to use. And so I think the challenge now is to do more experiments with the priority effects and then see whether the results are consistent with theory. And when theory fails, we can think about what happens. And one thing is that we can also have facilitative interactions that can lead to historical contingency. And that part is where modern coexistence theory is a little bit short of in what the theory can tell us.
Thanks. Another question coming from Nicholas, and he's asking about flower color on microbial community assembly, and especially, I guess, interactions with temperature and environment. So do darker flowers being warmer might show different patterns compared to lighter color flowers?
Yeah, good thought. Yes, I can see why color can make a difference because of the change in temperature. And so, for example, with bacteria, they seem to be less sensitive to temperature changes, fluctuation compared to yeast, and that lead to interesting interactions. For example, with we have another experiment led by Andrew Letton, who is now in Australia. When he was a postdoc with us, he looked at the temperature fluctuation and how the sensitivity of different species to temperature might have an effect on coexistence. And I can see when flower color affects temperature in nectar, that could have impact on microbial community assembly there. Another thing about flower color, obviously, is that the pollinator might also be affected by color. And so this classic idea of pollination syndrome, even though it's not as simple as the concept indicates, but if you have a red flower, it could be more of a hummingbird, a bird focused flowers, and then the other ones, like for example, yellow flowers, might be more of a bee pollinated flowers. So there are some studies showing that depending on the type of pollinators, the kind of microbial species that get to flower can be different. So that's another reason why flower color can make a difference to microbial communities in flowers.
Great, thanks. Next question from the wonderful Jacob Weiner. So he's thanking you for an excellent talk, but he's talking about plant community succession and that initial colonization can affect early stages of succession, but maybe not in the longer term because of how succession unfolds. And he asks, could this be because early colonists don't affect the environment as much as in heterotrophic systems like yours? So do you think autotrophic systems might be more resistant to long-term priority effects? That's
a very interesting thought. Yeah, I haven't thought systematically about the general difference between heterotrophic and autotrophic communities. I can see, yeah, why in autotrophic organisms like plants, maybe the kind of resources that are needed for them might be on average simpler, and so there might be smaller room for priority effects to affect communities, but we do have a lot of evidence showing that in plant communities too, particularly with they interact with the communities in soil, and plant soil feedback can lead to priority effects. So I'm not. I think it would be interesting to have some broad view with the existing data from different studies to see whether we have any systematic difference between heterotrophic and autotrophic. So my answer is that I'm not sure if the autotrophic communities may be less prone to historical contingency.
Yeah, I'm sort of thinking about you had a great paper on from New Zealand system on plant community change over time and showing there was some contingency, but maybe that's not quite in the long term.
Yes.
Okay, so I'm cognizant of time. I'm just going to ask one more quick question that I sort of flagged here before we go. And this is one I was thinking about as well as I was watching your videos. And this is about the dispersal between flowers. And so this person, is anonymous attendee, is asking about the role of wind and rain in dispersal and how to account for that. But I was thinking maybe more broadly also about biased versus non-biased dispersal in this system. So is there a bias in dispersal when we have pollinators that are actively selecting flowers? And is there other mechanisms of dispersal that might operate differently to interact with priority effects?
Yeah, great question. Yeah, in relation to wind and rain, as another way the dispersal can happen across flowers, we've done some experiments indicating that they do depend a lot on pollinators and other flower visiting animals for dispersal, but not entirely. And so some species we see, even if we exclude flowers from animal visits, indicating that there should be other ways like wind and rain, as this the person who asked the question. And another thing interesting thing we found is that it seems to depend how dependent the microbes are to pollinators for dispersal seems to be different. For example, some bacteria, we see them often, even if the pollinators don't come. Whereas if we look at the yeast, they seem to be more dependent on pollinators. And so that leads to some interesting questions about what those different species microbes can do to nectar chemistry. If you are not dependent on pollinators, you can change the nectar chemistry more freely without being affected, because if you're yeast and dependent on pollinators, you have to eventually move to another flower. Otherwise, that's the end of the population. And so you don't want to make the nectar so different so that the hummingbirds don't come. And so there is some interesting implication there. And then in terms of biased and versus unbiased disposal, that's also an interesting question. And conceptually, at least, something that I've been wanting to study experimentally. Conceptually, at least, you can imagine where there's a positive feedback so that once the plant becomes, or even the patches of plant become more or less yeast dominated, that's the favorite place for hummingbirds, and they keep coming back. And there is a well-connected network of flowers where it stays as the yeast-dominated plant or plant patch of plant. Whereas if you have a bacteria, then because hummingbird don't come to those plant or patches of plants, then there's a bias in dispersal, and that will become dispersal. So our experiments are focused on single flowers, but I think there is also a possibility of priority effect and alternative space, stable states at a larger scale, where entire population of plants could be, in some cases, going into either yeast or bacteria dominated. And so those are the thoughts that I have about bias dispersal.
No, that's great. Such a fascinating, interesting system. So that brings today's Ecology Live to a close. Thanks so much, Tad, for a wonderful talk, and thanks for everyone for joining in. And really sorry I couldn't get to all the questions, but Tad has agreed to interact with people on Twitter. You saw his Twitter handle at Tadashi Fukami. And you can use the hashtag Ecology Live to ask your questions and to continue this conversation. Thanks so much, Tad.
Thank you.
All right, so if you're enjoying the work of the BES, please get involved. We're currently recruiting for our committees and working groups who make vital contributions to our work and support the delivery of our vision for a world where nature and people thrive. There are a range of vacancies available, from our events committee to grants to publications and more. And we welcome applications from a range of backgrounds from all over the world. Find out about the roles and how to apply via the link in the chat. You should see it now. Make sure you apply before the deadline this Monday, the 19th of April. Next week, at the same time, we'll have Alex Moore from the American Museum of Natural History, who will introduce us to how our understanding of coastal wetlands is still far from complete. If you're watching the recording on YouTube, but would like to join for future live talks, make sure you register for Ecology Live on the events page of our website. Finally, I'll leave you with the details of an offer from the sponsor of today's Ecology Live talk, Oxford University Press. You can use the discount code on this slide and in the chat to receive a 30% off their Ecology Live reading list, which includes the book Floral Mimicry. Thank you again for attending, and see you next Thursday.